So over the last 25 years, we've developed methodologies that allow us to diagnose inherited conditions in human embryos that are produced during IVF. And this is called preimplantation genetic diagnosis. It's been really revolutionary for patients who want to avoid transmitting an inherited disorder to their children. It means that they can go through an IVF cycle, produce several embryos. Each embryo is tested. We find those that are unaffected by the condition, and those are the ones that are transferred to the patient. It means that the patient can begin day one of their pregnancy knowing that they will not have to contemplate the birth of an affected child or pregnancy termination. And that works really well, but there are still some problems with it. One of the issues is that some patients don't produce any embryos that are unaffected by the condition. Some patients may be opposed to pregnancy termination, but also opposed to the idea of discarding embryos that might contain the mutant gene causing the condition. So we've got interested now in the idea that maybe we should look not at diagnosis, but actually at cure of inherited conditions. And luckily, we have now available to us genetic tools that allow us perhaps to modify genes to correct mutations, to correct genetic defects, so that we could find affected embryos, correct the defect, and then have them transferred. This, of course, opens up the possibility that embryos could be rescued from an inherited condition, and that's a very exciting possibility.